Welcome dear students. Today we are going to learn about the dyes and tannins, their extraction, processing and use. The usage of natural colors is currently quite popular worldwide due to the carcinogenic and non-biodegradable nature of synthetic dyes. Natural dyes are the safest colors because they do not produce any unwanted byproducts and also contribute to the environmental regeneration. Undeniably, the natural dyes are healthier products purely because they do not comprise fitness-damaging chemicals. These dyes can be prepared without the use of manufacturing processes because they are extracted from nature. These colors do not harm the environment when they are used because they break down quickly in the nature. The primary applications for natural dyes are in the coloring of food, medicine, clothing and cosmetics. Paper, leather, shoe polish, wood, candles and other items needing coloring are also colored using small amounts of these dyes. Natural colorants cover all the dyes and pigments derived from plants, insects and minerals, hence derived from natural resources. The dyeing with natural colorants was one of the oldest techniques practiced by the people of ancient civilizations. This is evident from the Ajanta, Ellora, Sitanavasal, Mithila wall paintings and Egyptian pyramids, which had been exclusively done with natural colorants. India is incredibly blessed with a wide range of native plants. There are thought to be about 500 or more plant kinds in India that may produce natural colors. The nation's skilled artisans practice the art and craft of creating natural dyed textiles in numerous towns. When they use natural dyes, their color fastness and shade brilliance is somewhat limited. On the other hand, they yield quick and vivid results when combined with metallic mordants. The use of metallic mordants is not always eco-friendly. Therefore, instead of using violent technology for producing colors, one can use natural mordants, which is an eco-friendly way to achieve more similar results. Dear students, now we are going to learn about the classification of natural dyes. Based on structure, most commonly available natural colorants falling under this system of classification include indigoids, anthraquinones, alpha-naphthoquinones, flavones, dihydropyrins, anthocyanidin and carotenoids. Number 1. Indigoid dyes. This class is the oldest dye known to the world with evidence of its use stretching back at least 6,000 years ago. The Egyptian mummies buried in tombs were wrapped in the clothes dyed with indigo. Earlier, indigo was used as a black dye till its property to dye blue was discovered. The main coloring agent is a glycoside, namely indican, that occurs in the leaves of Indigofera tinctoria, which is a native to Asia, and several other species like Dyer's woad, Isatis tinctoria, which is indigenous to Europe, and Polygonum tinctorium. Next are the anthraquinone dyes. Quinonoids are fused benzonoid ring systems, 3 rings in case of anthraquinone and 2 rings in case of naphthoquinones, and possess enough conjugation to achieve color. Almost all the red natural dyes are based on the anthraquinoid structure, having both plant and mineral origin. Examples include Rubia tinctorium and Rubia cordifolia that have been used for dyeing textiles since 2000 BC. Mangistan and purpurine that are used to dye cotton, turkey red with alum. Imodin from Persian berries, and insect dye, cochineal or carminic acid from Cocos cacti produce brilliant scarlet on tin and alum, mordanted wool and silk. Number 3. Alpha-naphthoquinones. Typical example of this class is lawson or henna, cultivated mainly in India and Egypt. Another similar dye is juglone, obtained from the shells of unripe walnuts. These dyes are generally dispersing dyes and give shades of orange. Next are the flavonoids. Flavonoids are aromatic oxygen-containing heterocyclic pigments, specially located in the epidermal cells of plant parts and are chemically known as 2-phenyl-1,4-benzopyrene systems. They yield yellow dyes which can be classified under flavones, isoflavones, orones and chalcones. Most of the natural yellows are derivatives of hydroxy and methoxy substituted flavones and isoflavones. Common example is weld, containing luteolin pigment, giving brilliant and fast colors on both wool and silk. Next are the dihydropyrens, closely related in chemical structure to the flavones. These are important natural dyes for dark shades on silk, wool and cotton. Logwood, Brazil wood and sappanwood are the common examples. Next are the anthocyanidins. Anthocyanins are glycosides of 2-phenyl-benzo-pyroleum salts, intensely colored, sap-soluble plant pigments responsible for different colors in higher plants. These are probably the best known natural food colorants since they are widely distributed in the plant kingdom, and their color ranges from red of apples to green of grapes to purple of brinjals. The naturally occurring member of this class includes cerajurin, a direct orange dye for wool and cotton. It is obtained from the leaves of Bignonia chica. Next are the carotenoids. Linearly conjugated polyene tetraterpenes are represented by carotenoids, which are widely distributed in plants and animals. The carotenoids are brightly colored pigments in which the highly conjugated electron system confers different colors like yellow, orange, red to the molecule. Dear students, we will now discuss the extraction methods of natural dyes. Extraction of dyes from natural resources is a complicated process. They need specific techniques to extract dye from their original sources. The different techniques for extraction of natural colorants are: number 1, aqueous extraction. In this method, if the raw material is in dry form, it's broken into small pieces or powdered by grinding and soaked in water to loosen the cell structure. If the raw material is in wet form, it is then chopped into fine pieces or grinded into a fine paste. It's then boiled and is filtered to remove the non-coloring matters from the dye liquor. The aqueous extraction method has certain drawbacks, such as the need for a large amount of water, high temperatures, lengthy extraction times, reduced dye yield from the heat-sensitive coloring substances, and the ability to extract only water-soluble coloring components. Second extraction method is the alkaline or acidic extraction. The dye is extracted using an alkaline or acidic medium. Diluted acidic or alkaline solutions can also be employed for this, as they aid in the hydrolysis of glycosides and improve extraction. The natural dye, thisu, is extracted from the thisu flower using an acidic process. Phenolic groups in color extraction are extracted using an alkaline media. Using this technique, color is derived from lac insects, annatto seeds, and safflower petals. The third extraction method is ultrasonic microwave extraction. This technique involves treating raw materials in an aqueous solution with microwave or ultrasonic radiation in order to achieve high temperatures, short extraction times, and a faster rate of extraction. This process is used to extract color from annatto and butterfly pea seeds. Fourth one is the fermentation method. In this extraction technique, existing microorganisms in the raw materials are employed to carry out the fermentation and so aid the extraction process. This process extracts color from the seeds of annatto, indigo, and turmeric. Some drawbacks of this procedure are that it takes a lot of time, has an unpleasant odor due to microbial activity, and immediate extraction of color after harvesting is required. Fifth is the enzymatic extraction. Commercially available enzymes, including cellulase, amylase, and pectinase, have been used to loosen the binding material present in natural materials like bark, roots, or any hard part of plant. Next is the solvent extraction. Natural dyes are extracted using organic solvents such as ethanol, petroleum ether, acetone, and chloroform. The aqueous approach is less efficient than the solvent extraction method. Good quality dye yields are produced at low temperatures with little water requirements. Now we are going to discuss the supercritical fluid extraction. A fluid with physical characteristics halfway between a gas and a liquid, most commonly carbon dioxide, is used in this extraction to selectively extract target substances. Better contact with the substrate is achieved by its low surface tension, low viscosity, and good diffusivity qualities. Our next point of interest is the processing of the dyes. Once natural dyes have been extracted from the sources, the processing continues with several important steps to prepare the dye for application, optimize its properties, and ensure that it adheres well to the material being dyed. Processing involves the following steps. First is the filtration. The dye solution is passed through a fine mesh sieve, cheesecloth, or filter paper to remove plant particles, debris, and any other solid matter. Straining the boiled indigo leaves solution to remove leaf particles are an example. Number 2, concentration. In order to intensify the color of the filtered dye extract, the solution is further boiled to evaporate excess water and reduce the volume of the liquid. Alternatively, using a vacuum evaporator for a more controlled concentration process. For example, boiling down the strained madder root extract to concentrate the red dye. Third step is the mordanting. Mordanting improves the adherence of the dye to the fibers and enhance the color fastness. It can be done by pre-mordanting, treating the textile with a mordant solution before dyeing. Number 2, co-mordanting, adding mordants directly to the dye bath during the dyeing process. Number 3, post-mordanting, treating the dyed textile with a mordant solution after dyeing. An example to this is the soaking of cotton fabric in an alum solution before dyeing it with turmeric extract. Fourth step is the dyeing. Applying the concentrated dye to the material to impart the desired color. This involves wetting the textile to ensure even dye absorption, preparing the dye bath by diluting the concentrated dye extract with water, heating the dye bath to an appropriate temperature, immersion of the pre-wetted material in the dye bath, stirring periodically to ensure even coverage and prevent streaking, and soaking for 30 minutes to several hours, depending on the desired color intensity. An example to this is the simmering silk in a dye bath made from concentrated cochineal extract for a rich red color. Next step is the rinsing. Rinsing the dyed material in cold water until the rinse water runs clear, ensuring the removal of excess dye and any unattached particles from the dyed material. An example to this is rinsing wool dyed with indigo extract to remove excess dye. Next step is the fixing, which is optional. This is done to enhance color fastness and prevent fading. It involves vinegar, salt, or tannin solutions. The next step is drying. Drying the dyed material in a way that prevents color degradation, such as air dry away from the direct sunlight to prevent fading. Dear students, we are now about to explore the application of natural dyes. The natural dyes are generally applied in industries such as textile, food, leather, cosmetics, pharmaceuticals, etc. According to the literature, natural dyes such as walnut shells, onion shells, curcuma longa, Buteomonosperma, hibiscus rosa sinensis, Ziziphus jujuba, orange and lemon peels, Quercus infectoria, Albizia lebec, and so forth produce natural pigments and are used in textile dyeing industries. In addition to their biodegradability and compatibility with the environment, natural dyes have been recently discovered to exhibit other functional properties such as antimicrobial activity, antifungal, antiviral activity, insect repellent, UV protection, deodorizing agents, and dye-sensitized solar cells. Varieties of natural pigments like safflower, turmeric, quinines, anthocyanins, betalains, chlorophylls, carotenoids, tannins, etc. are used in coloration of foods. They have different groups of chemical compounds which may be used directly as colorants or in chemically modified form to produce different hues ranging from green through yellow, orange, red, blue and violet, depending on the source of colorant or increased stability. The cosmetic industry now employs many natural dyes due to the fact that they will cause fewer side effects than the employment of synthetic dyestuffs. But they can also provide extra properties such as UV protection, skin moisturizing and anti-aging. Natural pigments obtained from natural resources with active functional components have shown high biological activities and exhibited different properties depending on each kind of pigments, which make them excellent coloring materials for natural cosmetics and functional health foods. Students, let us throw some light on the mordants. Nearly all natural dyes require mordants to increase their substantivity. The majority of natural dyes need a chemical in the form of metal salts to create an affinity between the fiber and the pigment. These chemicals are known as mordants. Some of the important mordants used are potassium dichromate, ferrous sulfate, copper sulfate, zinc sulfate, tannin and tannic acid. It helps in absorption and fixation of natural dyes and also prevents bleeding and fading of colors, and thus improves the fastness properties of the dyed fabrics. There are three types of mordants, namely metal salts or metallic mordants, oil mordants and tannins. Our next area of focus is tannins. The term tannin is used for a range of natural polyphenols. It was first used by Seguin in 1796 and used to describe the process of transforming animal hides into leather by using plant extract from different parts of different plant species. Tannin is an astringent product found in a wide variety of plants. Plant parts include bark, wood, fruit, fruit pods, leaves, roots and plant galls. Tannin is defined as naturally occurring water-soluble polyphenolic compounds of high molecular weight, which is about 500 to 3000 Daltons, containing phenolic hydroxy groups to enable them to form effective cross-link between proteins and other macromolecules. Tannins do not denote a single compound. They include a large class of organic substances which often differ widely in their chemical composition and reactions. The tannins are divided structurally into two distinct classes, depending on the type of phenolic nuclei involved and the way they are joined. The first class is referred to as hydrolyzable tannins, while the other class is termed as condensed tannins. Now we are going to learn about the hydrolyzable tannins. These tannins are hydrolyzable by mineral acids or enzymes such as tannase. Their structures involve several molecules of polyphenolic acids such as gallic, hexahydro, diphenic or ellagic acids bounded through ester linkages to a central glucose molecule. Hydrolyzable tannins are sometimes referred to as pyrogallol tannins, as the components of the phenolic acids on dry distillation are converted to pyrogallol derivatives. These are obtained from myrobalan fruit, oak bark, gall nuts, etc. The hydrolyzable tannins are soluble in water and their solution produces blue color with ferric chloride. Now we are going to discuss the condensed tannins. Condensed tannins are not readily hydrolyzable to simpler molecules with mineral acids and enzymes. Thus, they are also referred to as non-hydrolyzable tannins. The term proanthocyanidins is sometimes alternatively used for these tannins. The compounds containing condensed tannins contain only phenolic nuclei, which are biosynthetically related to flavonoids. Condensed tannins are also soluble in water and produces green color with ferric chloride. Next is the sources of tannins. In the same plant, different parts like bark, nut, leaf and wood may contain different types of tannins. The families of the plants rich in both groups of tannins include Rosaceae, Geraniaceae, Leguminosae, Combataceae, Rubiaceae, Polygonaceae, Thioaceae, etc. The members of families Cruciferae and Papaver, on the other hand, are totally devoid of tannins. In the plants in which tannins are present, they exert an inhibitory effect on many enzymes due to their nature of protein precipitation and therefore contribute a protective function in barks and heartwood. Some of the plant species containing tannins are gallnuts having 50 to 70% tannins, myrobalan having 30 to 35% tannins and chestnut having 30% tannins. Extraction of tannins. Due to heterogeneous nature of tannins, the extraction process remains as the main challenge for their valorization. The extraction of tannins is not done in a single protocol and the procedures are widely variable. Because of the extraction process, the extracted tannin contains different types of impurities including minerals, stilbenes and sugars. For instance, the presence of carbohydrate in tannins reduce its antifungal efficacy as well as create problems during impregnation into wood for preservation. Additionally, the number of impurities depends on the processing parameters like particle size, temperature, pressure, time, solvent type and solid to solvent ratio. In general, tannins are extracted from the plant materials such as bark, wood, stem, leaf, either by hot water or water along with other solvents. Acetone, diisopropyl ether, ethyl acetate, ethyl ether, methanol, ethanol, sodium sulfite and sodium hydroxide has been used by different researchers as solvent with or without water. Acetone has been used to extract tannins from dried leaves and green stems of rock rose, whereas 75% methanol with water was used to extract tannins from pine barks. Microwave technology used to extract tannins from black wattle barks where water, 80% methanol and 80% ethanol were used for infusion and maceration. Besides, 60% methanol has also been used with ultrasound technology to extract tannins from acorn. Sodium hydroxide was also used for tannin extraction from Missouri berry, and 0.22% concentration among the different sodium hydroxide concentrations showed the best results. It is also reported that methanol extraction provided more tannin yield compared to the water extraction. Addition of additives like enzymes such as polygalacturonase and pectin lyase in the solvent positively influence the quality and amount of tannins. However, among the different methods, hot water extraction method is still popular and commonly used for the extractions of tannins in the industry as well as in the laboratory. The simplicity of the method and its lower cost are the main reasons for its popularity. Moreover, some of the researchers even reported that the amount of extracted condensed and hydrolyzable tannins were the highest when the solvent was hot water. Depending on the solvent type, this temperature can be as high as 120 degrees Celsius. Even with water as solvent, higher temperature of almost 105 degrees Celsius was used when tannins were extracted from red and jaboticaba and imbu barks. When methanol or ethanol was used with water as solvents, the temperature ranged between 60 and 120 degrees Celsius. However, higher temperature showed better results. Pressure application helps to extract tannins at lower temperature, as well as the advanced technologies like ultrasound, microwave technology require lower processing temperature. For efficient extraction, these plant parts are grounded prior to any type of extraction. However, the particle size varies depending on the parameters like efficacy level, time, temperature, solvent, raw materials, and process. Besides the aforementioned tannin extraction techniques, there are many other advanced techniques, including supercritical ionic liquid assisting microwave, infrared assisted and pressurized hot water extraction technique. In the pressurized water extraction method, 374 degrees Celsius temperature and 22.1 megapascals pressure are used. In infrared technique, infrared lamp is used for heating during extraction. The efficiency of this method depends on the wavelength of the infrared heater, distance between source of infrared and material, and the solvent. Supercritical is another modern technology for extracting tannins, where supercritical solvents such as carbon dioxide, butane, pentane, fluorinated hydrocarbons, sulfur, hexafluoride, and nitrous oxide are used. The extraction efficiency and the quality of the tannins depend on the duration of the extraction. Hence, long extraction duration yields higher amount of tannins. The cell structure of plant is destroyed with time in the solvents, and yield of tannin extraction is increased. However, quality might decrease with time. Solvent to solid ratio is another important factor which governs the tannins extraction process. We will now discuss the processing of tannins. The processing of tannins after extraction involves several steps to purify, concentrate, and prepare the tannin extract for its intended applications. Removal of solid residues from the tannin extract to obtain a clear solution using a fine mesh sieve, cheesecloth, or filter paper to strain the solution. For example, straining the oak bark tannin extract to remove bark particles. Increase the tannin content by reducing the volume of liquid by boiling the filtered tannin solution to evaporate excess water. This includes boiling down the strained tannin solution from chestnut wood to make it more concentrated. Next is the removal of unwanted impurities and non-tannin substances by precipitation, using a substance that precipitates impurities out of the solution such as lime or aluminum sulfate, and then filter again. Next step is the standardization that ensures consistent quality and concentration of tannins in the final product using chemical analysis methods such as spectrophotometry or titration. Lastly, we will discuss the application of tannins. Number 1. Tannin in leather processing. Leather making is a very old craft that dates back to 1500 BC. Through the use of tan liquor, which provides increased stability and resilience to heat, water, bacteria, and abrasion, tannins are used to turn animal hide into leather. Leather is created during this tanning process, when tannin cross-links the collagen chains in the hide. This is because the different combinations of tannin protein polymers occur. The major contributors for the world's supply of condensed tannins are extracted from mangrove species, wattle and quebracho. Condensed tannins are the major component of the commercial tannins, which are used for tanning of leather in a tannery. Condensed tannins have high resistance to detannage, thus it is suitable for tanning of leather in the industry. Therefore, it controls the bacterial growth for avoiding bad odor and makes stronger leather. The binding of tannins with collagens is dependent on temperature, pH, and molecular weight of tannin. Number 2 is the tannins in textiles. Tannins are used as mordant in dyeing, manufacture of ink, sizing paper, and silk, and for printing fabrics. Tannins are significant in textile dyeing for several reasons. Firstly, they produce indispensable mordants for the dyeing of vegetable fibers like cotton and linen. Secondly, they are very often associated in the plants with the yellow, orange, red and violet colorants, whose color is reinforced by them in the dye bath by their own pigment. Materials dyed with tannins have good fastness for washing and light. Third use is the tannins in medicine. Most of the Indian systems of medicine, such as Ayurveda, Siddha and Unani formulations, contain many types of tannins as ingredients. Tannins are astringents in nature and used in Ayurveda and Siddha tooth powder in order to strengthen the gums. They also promote rapid healing and the formation of new tissues on wounds and inflamed mucosa. Tannins are used in the treatment of varicose ulcers, hemorrhoids, minor burns, frostbite, as well as inflammation of gums. Internally, tannins are administered in cases of diarrhea and in cases of heavy metal poisoning as an antidote. In recent years, these compounds have demonstrated their antiviral activities and for treatment of viral diseases including AIDS. Number 4. Preparation of plastics and adhesives. Condensed tannins are phenolic raw materials which react with formaldehyde, and these can be used for the production of synthetic resins. Tannins help to make greater adhesion between fibers and matrix and achieve better physical and mechanical properties of the wood-based composites. Number 5. Anti-corrosive chemical for metals. Tannins protect the iron materials from sulfate-reducing bacteria by exerting a bacteriostatic action. Mangrove tannins are economically viable to protect the underground iron pipes and tubes. Condensed tannins have flavon-3-ol, which has oxygen atoms in its functional groups and aromatic rings, and aromatic rings can scavenge oxygen and inhibit corrosion. Thank you all for today's lecture. I hope you found it informative and engaging.